guys, so today we'll be talking about Clustal Omega. Before we start, I think it's important to talk about multiple sequence alignment. Sometimes scientists like to see the similarities between DNA strands encoding for genes, proteins, etc. And they want to figure out the best alignment for it, or basically a quantification of similarity. So we have our original gene right here. We're comparing it to this one. There's a gap, so these two don't match. These match, two T's, two C's match. G and A do not. So that means that this is about, out of 4, we only have 4, whereas scientists would have many more than this. We have 4 right here, and it's pretty much only 50% similar. So that's really not enough to really identify or be able to use the data for comparison. It's like a waste of time, essentially. So if that happens, scientists, like I said, they do a multiple alignment. So they'll take the whole entire orientation of the sequences and move them to the left or right until they get the most similar alignment overall. This is going to be our starting gene again. As you can see for this one, 3 of them don't match. And then the G is a gap. So this one is not aligned at all. This one is also not going to be acceptable to use because it does have one alignment right here, but the other 3 are not. So the dilemma we have is, how do scientists quantify this? Or what's the limit at how similar they are, and when we can use the similarity to make judgments or observations of similarities between certain genes, mutation, etc. that the biologists can be looking at. And the answer to that is they use a marked distribution system. So in the marked distribution system, if there's a match, there's a +2. It's like basically a grading system. If there's X, -1. If there's nothing, it's going to be -2. So they do this for every base pair, and then the computer adds up all the base pairs, and it gives you a total number. And the more aligned that the sequences are, the higher the number is going to be, because they're going to have more greens. And that is the most compatible sequence to our original sequence, as well as to any other sequences that are acceptable based on their quantifications for similarity. Why do biologists use sequencing alignment? So biologists compare sequence similarity to identify homologous genes. Homologous genes are shared genes over time from common ancestors, but over time they acquire mutations, insert deletions that alter the DNA, which then alter the effects that that sequence has on the living being. 2 genes are homologous if they share a common ancestor. Therefore, homology is a dichotomous characteristic. But if you're giving 2 genes based on their sequence, there is more of a spectrum of how homologous they are. It breaks it down so we can look at those small differences that can create new species, new diseases, new genes that could help regulate things or take certain things out. And why do we want to find these genes? Because we can identify new genes. Identify all open reading frames of their genome. The links can be used to estimate the probability of constituting genes. On the other hand, if there's a gene from another organism with a good alignment with the ORF found, there is a strong evidence that the ORF is a gene. Protein modeling. So similar sequences often have similar to secondary tertiary structures. It's very useful in comparing the structures with the DNA. That's compared to other species, how the structures differ based on how the DNA sequences differ. Function prediction. The alignment of sequences to determine if 2 genes are similar. If the first function of these genes is known, we can assign the same function as the second. So we have our original gene. We know this part, it creates like a specific function in the body. And if the first function is similar enough to the next gene, then they'll both have that function. Or if they're 2 different, then that's something to note, that they don't function the same way because of the sequences and the differences that we see by aligning them. So biologists choose this database to find sequence similarities compared to other methods because it's hard to determine what part of this DNA is exon or intron, especially in animals. But we know based on sequencing alignment that there's always a start codon and a stopping codon for an exon. So the start intron is usually the same ATG. The stop, you can get 3 different triplets there, and basically what this does is it helps us identify where these ATGs are, like where approximately the length of exons in comparison to other sequences, and the insinuations behind that. So as far as the database goes, I'll go ahead and open up my browser really fast. So basically you can input your gene in any supported format. You can click on formats and it gives you a gigantic list of formats that you can use, and it has explanations for you of how to put them into this box. I'm going to use an example sequence. Oh, this is it. So as you can see, there's so many different ways that you can put nucleotides, proteins into this. It has a whole table of formats and how it looks, which is really nice. So we could take this gene here, for example. Copy. We'll see how this is. It's, what is it called? FASTA. And then I usually just click the default. You definitely have to look up more information about these output formats in terms of how they will be useful to you based on what you're researching. You can be notified via email if you have big sets of data. Let's see. It's running. Okay. And here's the results of our job. So basically, what we are looking at here is these are all these sequences that it found that had the alignment score that was high enough to determine that it was significant for scientists to determine whether we can use it to look at similarities or differences among genes and record information based on their expressions. So these are the specific sequences. It outputted it for us. This encodes for most likely a gene, and as you can see how we talked about earlier, this starts with ATG, starting codon, so we know that one is a exon. Some of these sequences don't have ATGs or start or stop codons, so that could be indicative of, oh, there's a stop codon at the bottom. Okay, so this is a whole part of a exon, the whole section of exon, which is really, really nice. The computer automatically took out the introns for us. So we can just look at direct comparisons here in the similarities and look at numerical scores and determine how significant they would be in us utilizing. There's a couple things up here that could possibly be useful. Like the phylogenetic tree, you can look at it between these 4 genes that we saw on the first page and see that these 2 are more closely related than these two over here. But other than that, this section only really is useful for JAL view. So usually I would click on JAL view, but it is a software program that takes a while to boot up. So I have a couple screenshots from a different example I use. I believe this is ferredoxin, which is a gene that exists in spinach, I believe. I'm not exactly sure. But this shows you a colorful, the colors are so helpful in seeing, like, these are straight lines of colors, so you know straight lines would indicate that this is very similar between all of them. And the more orderly the lines are in certain sections, you can see which sections are more similar versus which ones aren't. And they have these quantities down here, conservation. So the conservation of adenine was throughout all of these genes. So the conservation bar is going to be really high. As far as these genes, these vary significantly. So it's going to have a low bar right here. All of these, where the straight lines are, it's going to have a really high bar right here for conservation. And then the consensus is the merging between the quality and conservation of the gene. As far as protein mapping goes, this is not a useful program for that, considering there are many other free programs that we have that could do a better job at letting us look at the different parts of the protein. This just draws a basic structure. You can't change and look at different structures in here. You can change colors, you can rotate it around, but it's just not really good graphic quality. And again, there are many other databases that we can use other than this. Another useful thing that I found, I thought was really interesting, it puts it in a pictorial perspective. Again, talking about homologous genes, all of these species share this homologous gene with this species. But over time, because of mutations and such, we have these branches that are divergent of each other that carry different genes. So these two would be more similar than these two or these two because they have the closest latest common ancestor, whereas these guys would be less similar than or this guy would be less similar than these guys. And that's pretty much all that this database can offer. I think it's really useful mainly for its multiple sequencing alignment and this chart, which could be excellent for poster presentations and research on the gene in general.